Welcome to this lecture on the fundamentals of genetics. Today, we are going to explore a fascinating topic called organelle inheritance. When we think of genetics, we usually picture the DNA tightly packed inside the nucleus of a cell. But what if I told you that there is a whole other set of genetic instructions living outside the nucleus, playing a massive role in your health, evolution, and even the food we grow? Understanding this hidden genetics is crucial for modern biology. Let us dive in and discover how these unique traits are passed from one generation to the next. Now that we know what our topic is, let us start with a simple analogy. Imagine you are building a brand new house. You receive a massive, detailed blueprint that is an equal mix of ideas from both of your parents. This represents the DNA inside the nucleus of a cell. However, a house also needs independent power generators to keep the lights on. In human biology, these power generators are the mitochondria. Interestingly, these generators come with their own tiny, separate instruction manuals. Organelle inheritance is the study of how these specific tiny manuals are passed down from generation to generation, breaking the standard 50-50 rule of genetics. Building on that house analogy, let us look at the biological terms for this process. Organelle inheritance is also known as extranuclear or cytoplasmic inheritance. This is because it involves the transmission of genes that are located entirely outside the nucleus of the cell. Specifically, these unique genes reside within the circular DNA of cytoplasmic organelles. In animals, these are the mitochondria, and in plants, this includes both mitochondria and chloroplasts. These organelles act as independent entities within the cell's cytoplasm, carrying their own genetic codes separate from the main nuclear genome. Now that we know where these genes are located, how are they passed on? Unlike nuclear genes that follow standard Mendelian inheritance patterns with a 50-50 split from both parents, organelle genes play by different rules. They typically exhibit uniparental inheritance. In most eukaryotic organisms, this translates to strict maternal inheritance. Why does this happen? It is because the female gamete, the egg, is massive and provides all the cytoplasm to the new zygote. The male gamete contributes essentially only a nucleus, meaning the father's organelle DNA is completely bypassed. To truly grasp why this inheritance is strictly maternal, let us look at this diagram of fertilization. On the left, you see the female gamete, or the egg. It is massive and packed with cytoplasm, which includes numerous red mitochondria. Approaching it is the male gamete, the sperm, which is essentially just a nucleus with a tiny amount of cytoplasm and a few blue mitochondria for propulsion. When they fuse to form the offspring cell on the right, the egg provides all the cytoplasm. Notice what happens to the blue paternal mitochondria. They are actively degraded and destroyed. Therefore, the new offspring cell inherits its power plants exclusively from the mother. Now that we understand the mechanism of maternal inheritance, let us explore why it is so critical in the real world. In modern medicine, mutations in mitochondrial DNA can lead to severe metabolic disorders. For example, Leber's hereditary optic neuropathy primarily affects energy-hungry tissues like the brain and muscles. Because these diseases are strictly maternally inherited, genetic counselors can predict risks with certainty, knowing an affected father will never pass the disease to his children. In agriculture, plant breeders exploit a trait called cytoplasmic male sterility, governed by organelle genes. This prevents crops like corn from self-pollinating, allowing farmers to efficiently produce high-yielding hybrid seeds without the expensive process of manually removing pollen. This brings us to an important cellular reality regarding these diseases. Because a single cell contains hundreds or even thousands of mitochondria, they might not all share the exact same DNA sequence. Sometimes a cell ends up with a mixture of perfectly normal organelles and mutated ones. We call this mixed state heteroplasmy. Understanding heteroplasmy is vital because it determines how likely a cell is to actually show symptoms of a mitochondrial disease. If the proportion of mutated organelles is low, the healthy ones can compensate. But if the mutated ones multiply and cross a certain threshold, the cell's energy production fails and disease symptoms appear. To quantify this risk we just discussed, we use a simple mathematical formula to calculate the heteroplasmy ratio. Conceptually, this formula just finds the percentage of a cell's power plants that are broken. Here, H represents the heteroplasmy ratio, which is the decimal fraction of mutant organelles in a single cell. M-mut stands for the number of mutant or defective organelles present. MWT represents the number of wild-type or perfectly normal organelles. The denominator simply adds them together to get the total number of organelles. If the number of mutant organelles increases, the ratio H increases, pushing the cell closer to the disease threshold. Conversely, if wild-type organelles replicate faster, H decreases, protecting the cell. Before we move on, let us see this formula in action with a concrete medical example. A patient is tested for a mitochondrial disease causing muscle weakness. We are given that out of a total of 5,000 mitochondria, exactly 3,500 carry the mutation. The critical disease threshold is 0.60. We need to find the patient's ratio and predict if symptoms will appear. We choose our heteroplasmy formula because it directly compares mutant count to the total count. We substitute 3,500 for the numerator and 5,000 for the denominator. Dividing these gives a final ratio of 0.70. Because 0.70 is strictly greater than the threshold of 0.60, the defective power plants overwhelm the healthy ones, meaning the patient will physically exhibit severe muscle weakness. Now that we have successfully calculated mitochondrial inheritance in humans, let us shift our focus to plants and algae, which also have chloroplasts. Chloroplasts contain their own DNA and follow similar rules of organelle inheritance. In the single-celled algae Chlamydomonas, chloroplast inheritance is determined by specific mating types, designated as MT+ and MT-. Nature has designed a strict uniparental system here. The MT+ parent transmits its chloroplast DNA to 100% of the offspring. Meanwhile, the MT- parent transmits absolutely nothing. Its chloroplast genetic contribution is completely ignored and lost in the next generation. To predict exactly how traits are passed down in these algae, we use a simple transmission probability formula. Conceptually, this just multiplies the total number of children by the chance that the dominant mating type passes on its genes. Here, N-res stands for the expected number of offspring that will inherit the specific chloroplast trait, like antibiotic resistance. N-total is the total number of offspring produced by the mating event. Finally, PMT+ represents the probability of the MT+ parent passing on its chloroplasts. Because this inheritance is strictly uniparental from the plus mating type, if the trait is in the MT+ parent, this probability is exactly 1.0. If the total offspring increases, the number of resistant offspring increases proportionally. Let us apply this transmission formula to a specific genetic cross. We are given that a streptomycin-resistant MT+ algae mates with a sensitive MT- algae, producing exactly 400 offspring. We are asked to calculate how many offspring will be resistant. We use our transmission formula because resistance is a chloroplast trait. First, we identify that the resistant trait belongs to the Mt plus parent. Therefore, the transmission probability is a guaranteed 1.0. We substitute 400 for the total offspring and multiply by 1.0. The final answer is 400. Physically, this means every single one of the offspring will survive exposure to the antibiotic, perfectly illustrating how the sensitive trait from the Mt minus parent is completely erased. We have covered a lot of ground today, so let us summarize the key takeaways. First, organelle inheritance fundamentally shifts our understanding of genetics by proving that not all traits are inherited equally from both parents. Mitochondria and chloroplasts have their own DNA. Second, these vital energy-producing organelles are passed down through strict uniparental, usually maternal, lines. Third, we learned that the balance of normal and mutated organelles, known as the heteroplasmy ratio, dictates whether severe metabolic diseases will appear. You now have a solid grasp of how this unique mechanism traces evolutionary lineages and even helps manipulate crop genetics. Excellent work mastering this complex topic.